So I want you to imagine that you are competing in like this elite triathlon.
Oh, wow. Okay.
Yeah, right. You have trained for months. You are in absolutely peak physical condition.
You're feeling invincible.
Totally invincible. The starting gun goes off. You dive into this pristine, cool, freshwater lake, and you just power through the swim.
Sounds incredibly refreshing.
Right. It's restorative. You finish the race, you go home, and you feel great. But then about 2 weeks later, you are lying in an intensive care unit.
Oh, wow. That escalated quickly.
It really does. Your kidneys are shutting down. Your skin has turned this deep bronze yellow color.
Terrifying.
And most terrifyingly, your lungs are inexplicably filling with blood. And your doctors, they have absolutely no idea why.
Yeah, I mean, that is the ultimate medical nightmare right there. You were literally dying from an exposure you didn't even realize happened.
Exactly.
And the clock is just ticking way faster than the diagnostic tests can even run.
Well, that's exactly what we are getting into today. Welcome to the deep dive. We are exploring one of the most widespread, yet, significantly underdiagnosed zoonotic infections on the planet today.
It really is a biological booby trap.
That's the perfect way to put it. It goes by all these vague nicknames like swamp fever or mud fever.
Or Wheels disease.
Right, Wheels disease. But its actual scientific name is leptospirosis.
Leptospirosis, yeah.
And I have to say, the statistics we pulled from this massive stack of clinical guidelines and epidemiological reviews and, peer-reviewed case studies for this deep dive, they are just staggering.
They really are. I mean, the numbers are huge.
We are talking about over 1 million cases and roughly 60,000 deaths annually worldwide.
Which really brings up the central mystery that we need to unravel today.
Right.
How does a disease that is entirely curable with basic, readily available antibiotics somehow manage to kill 60,000 people every single year?
That is the exact question driving our mission today. We want to give you, the listener, a total shortcut to being deeply well-informed about this extremely stealthy pathogen.
Absolutely.
We are going to figure out how it kills by looking at how it hides right in plain sight.
How it literally just walks right past the human immune system.
Yeah, how it disguises itself as dozens of other diseases, and crucially, why extreme weather and climate change are turning it into a massive threat for just about everyone.
From urban residents to those adventure athletes we were just talking about.
Exactly. So to understand how this pathogen operates and why it's so incredibly difficult to catch, we actually need to rewind the clock a bit.
Yeah, we have to look at the historical puzzle it presented to the very first doctors who even tried to define it.
Because for a really long time, it wasn't a disease with a name.
No, it was just a terrifying cluster of symptoms.
Right. So we are talking about Germany back in 1886.
Yes, 1886. There was this German physician named Adolf Weil, and he started noticing a very specific, just very severe clinical syndrome in his hospital.
What kind of patients was he seeing?
He was observing patients who were mostly coming from outdoor, heavily water-exposed occupations.
Like who?
Think coal miners, right? Guys working in these flooded subterranean shafts or sewer workers.
Okay, that makes sense.
Yeah, and they were coming into the clinic with this sudden acute infectious disease that just progressed so rapidly.
And the symptoms he documented were incredibly dramatic, right?
Absolutely devastating.
He's writing about patients with massively enlarged spleens, severe jaundice, which is, that deep yellowing of the skin and eyes.
Right.
And nephritis, which is basically profound inflammation and total failure of the kidneys.
It was awful. But the frustrating part for Adolf Weil, and really for the whole medical community at the time, was that bacteriology was still in its relative infancy.
They just didn't have the tools.
Exactly. They could see the damage, they could categorize the syndrome, but they literally could not find the culprit.
So for decades, this specific severe presentation just bore his name, Weil's disease.
Yeah, they knew it was infectious. They suspected it was tied to contaminated water or mud somehow.
But the actual bug remained completely invisible.
Completely invisible.
And it stayed invisible until about 1907. And the way it was finally spotted was, it was essentially an accident.
An accident, yeah.
There was this researcher named A.M. Stimson.
Right.
And he wasn't even looking for Weil's disease at all. He was examining kidney tissue from a patient who everyone just presumed had died of yellow fever.
Which was a massive scourge at the time.
Exactly. So he's doing this post-mortem tissue analysis.
But when he actually prepared the kidney tissue and looked under his microscope, he didn't find the viral pathology of yellow fever at all.
What did he see?
Instead, He saw these bizarre, tiny microorganisms embedded right in the tissue.
Just hanging out there.
Yeah, and they didn't look like standard round or rod-shaped bacteria either.
Not at all.
He named the organism Spirochaeta interrogans.
And that second word, interrogans, that is the key, isn't it? Because of what it actually looked like under the lens.
It looked exactly like a question mark.
A microscopic question mark. I love that.
Right. The organism had this very peculiar, tightly coiled shape with these distinct hooked ends.
Like it was literally interrogating him from the glass slide.
Exactly. And then a few years later, in 1917, a prominent bacteriologist named Hideo Noguchi looked at it.
Okay, what did he find?
He realized this wasn't just a standard spirochaete. He officially proposed a brand new genus name for it.
Which was Leptospira.
Yes, it combines the Greek word leptos, which means fine or thin, and the Latin word spira, meaning coil.
A fine, thin coil.
Exactly.
You know, when I was going through the microbiology notes in our sources, just trying to visualize this thing, I honestly couldn't stop picturing it as a microscopic drill bit.
That is a highly accurate way to picture it, actually.
Because it's categorized as a flexible, spiral-shaped, gram-negative bacteria. But the way it moves, that is what really caught my attention.
It is unique for sure.
Because it doesn't just float around in the bloodstream like some passive passenger.
Not at all. It is highly, highly modal, and its method of locomotion is just aggressively invasive.
How does it move compared to normal bacteria?
Well, most modal bacteria use flagella, which are these whip-like appendages that stick out from their back end.
Right, like a little propeller on the back of a boat.
Exactly, like a tiny boat motor. But Leptospira is completely different. It possesses two paraplasmic flagella.
Okay, what does that mean?
These are axial filaments, essentially internal motors, anchored at each pole of the bacteria. They run along the inside body of the cell underneath its outer sheath.
Wait, so instead of having a propeller pushing it from behind, the entire body of the bacteria is the motor.
That's the perfect way to visualize it.
That is wild.
Yeah, when those internal filaments rotate, they cause the entire bacterial body to twist in this aggressive, corkscrew-like rotational movement.
Just drilling.
It gives the bacteria both rotational and translational movement. And this is a massive evolutionary advantage.
Why is that?
Because it allows the bacteria to literally bore its way through highly viscous environments.
Like human tissue.
Exactly. If you put a normal propeller-driven bacteria in thick connective tissue, it just gets bogged down and stuck.
It can't swim through the mud, basically.
Right. But Leptospira, it just corkscrews right through the cellular matrix.
Wow. And the sources say it's an obligate aerobe, so it needs oxygen, and it thrives in warm temperatures, like around 28 to 30 degrees Celsius.
Warm and wet, yes.
And it is insanely small.
It is only about 0.1 micrometers in diameter.
Which is hard to even fathom.
To put that in perspective, that is thin enough to just slip right through the tiny microscopic spaces between our own human cells.
Spaces that are normally completely sealed off.
Completely impermeable to other microbes, yes.
Okay, now, you mentioned earlier it's a gram-negative bacteria. For those of us who aren't microbiologists, what does that actually mean for the patient? Like, why does that matter?
It's a great question because it really dictates how hard this bacteria is to kill. In the bacterial world, there are generally two types of cellular armor. You have gram-positive and gram-negative.
Right.
Gram-positive bacteria have a thick, single-layer mesh wall. But gram-negative bacteria, like Leptospira, they have a dual-layer system.
The dual layer.
Yeah, they have a thin inner wall, but then they have this incredibly tough, highly protective outer membrane.
So it's like they're wearing a heavy suit of armor over a chainmail shirt.
That is exactly it. And this outer membrane protects them from many of the host's natural defensive enzymes.
Making them harder for our bodies to fight.
Right, and it makes them inherently resistant to certain entire classes of antibiotics.
Okay, so we have this microscopic armor-plated drill bit.
Yes.
But reading the literature, trying to figure out what to actually call this specific drill bit, gave me a massive headache.
Oh, the taxonomy, yes.
The medical world seems to be speaking two completely different languages to describe the exact same biological invader.
It is notoriously confusing.
There is this serological classification system, and then there is a genomic classification system. Why is it so complicated?
It frustrates clinicians too, believe me. But we really have to understand both because they answer two fundamentally different questions.
Which are?
How does our body see the bacteria versus how dangerous is the bacteria itself?
Okay, break that down for me.
Let's tackle the serological system first.
Okay.
This is the older, more traditional system. It classifies the bacteria based on the antigens on its surface.
The outside of the armor.
Exactly. Specifically its surface lipopolysaccharide, or LPS.
When I was reading about this, I found it helpful to think of the LPS as the bacteria's external uniform.
That's a brilliant analogy. It's the uniform the bacteria wears on the outside of its armor.
Okay.
And based on these tiny microscopic chemical variations in this uniform, scientists have identified over 300 different pathogenic serovars.
300 different uniforms.
Yes, you'll see these names in the literature all the time. Serovar icterohemorrhagiae, Serovar canicola, Serovar pomona.
And the uniform is what our human immune system actually sees and reacts to.
Yes, exactly.
So if I get infected with, say, Serovar canicola, my immune system learns to recognize that specific uniform.
Right. It builds defensive weapons, specific antibodies, against the canicola uniform.
Which is vital for diagnostics and vaccines, I assume.
It is entirely vital. Vaccines are almost completely Serovar specific.
Oh, wow. So if you are vaccinated against the canicola uniform,
your immune system is totally ready for it. But if a Leptospira bacteria wearing the pomona uniform shows up, your immune guards might just let it walk right by.
Because they just don't recognize the outfit.
Exactly. They're looking for a blue shirt, and this one is wearing red.
But then we have the genomic classification.
Right. Modern science eventually allowed us to look past the uniform and sequence the actual DNA.
The genetic blueprint of the bug.
Exactly. And when we looked at the blueprints, we found around 68 to 70 distinct species of Leptospira.
Okay.
These species are grouped into clades based on their inherent virulence, meaning how dangerous they are.
Right.
So the pathogenic P1 clade contains highly dangerous species like Leptospira interrogans.
And the intermediate P2 clade causes, like, milder disease.
Correct. But the really confusing part is that the uniform doesn't always match the blueprint.
Wait, really?
No, it doesn't map neatly at all. You can have the exact same Serovar uniform appearing on bacteria that have completely different genomic blueprints.
Oh, man. So to summarize this for the listener, just to keep it straight.
Please do.
The Serovar, which is the uniform, tells us how our immune system and our vaccines will react to the surface of the bacteria.
Right.
But the genomic species, the blueprint, tells us the pathogen's evolutionary virulence, meaning how inherently dangerous its internal machinery is once it actually gets to work inside us.
Perfect summary.
Okay.
And knowing that duality is totally crucial for researchers trying to track outbreaks and develop better treatments.
Which brings us to a really uncomfortable question, I think.
Okay.
We know this pathogen is a tiny armor-plated corkscrew designed to burrow through tissue. How does it actually find its way into our bodies in the first place?
The route of transmission.
Because we need to look at the reservoir. Humans are not the main character in this bacteria's story, are we?
Not at all. We are barely even a footnote in its natural life cycle. Leptospirosis is a zoonotic disease.
Meaning it comes from animals.
Right. Its natural life cycle happens entirely within animal populations. Specifically, the bacteria colonize the proximal renal tubules.
The kidneys.
Yes, the microscopic tubes inside the kidneys of what we call maintenance hosts.
And the undisputed king of the maintenance hosts globally is the brown rat, right? Rattus norvegicus.
The brown rat is the primary vector in urban settings, absolutely.
Mm-hmm.
Though the sources note that dogs, cattle, pigs, and even horses can also serve as reservoirs.
But the rat is the perfect vessel. And this is where the biology gets truly insidious, honestly.
It really does.
When the bacteria colonize the rat's kidneys, the rat doesn't get sick. It is completely asymptomatic. Wait, how is that even possible? We just spent 10 minutes talking about how aggressive and destructive this bacteria is. Why doesn't it just destroy the rat's kidneys?
It comes down to a really fascinating evolutionary truce.
A truce.
Yeah. The rat's innate immune system, specifically a receptor called TLR4, which we will definitely discuss in depth shortly.
Oh, we will get into TLR4.
Well, the rat's TLR4 perfectly recognizes the Leptospira bacteria.
Okay, so it sees the bug.
Right. And the rat's immune system keeps the bacterial population strictly confined to the kidneys. It doesn't let the bacteria invade the rest of the organs.
But it also doesn't kill it.
Exactly. It can't completely clear it from the renal tubules. So they basically reach a stalemate.
Wow.
The bacteria set up a permanent camp in the kidneys, and the rat just goes about its life feeling totally fine.
While shedding millions of active bacteria in its urine every single day.
Every single day for months or even for its entire lifespan.
So we have all these asymptomatic rats running around, urinating everywhere. What happens to the bacteria once it leaves the rat?
Well, if that urine hits, say, dry concrete in the hot sun, the bacteria dies pretty quickly.
Okay.
It is highly susceptible to desiccation or drying out. But if that urine contaminates a favorable environment,
And what's favorable?
warm, moist, neutral to alkaline fresh water or mud, if it hits that, the leptospires can survive outside a host for weeks or even several months.
They just wait.
They just wait in the water.
It's essentially a biological landmine left behind in a puddle or a lake or a flooded street.
Exactly.
And the medical literature refers to humans as incidental or dead-end hosts.
Right, because the bacteria isn't actively trying to infect us to complete its life cycle.
It doesn't need us, and person-to-person transmission is unbelievably rare, right?
Exceptionally rare. You essentially have to stumble into the contaminated environment yourself.
But I have to ask, how does it actually breach our defenses?
What do you mean?
Like, you're just swimming in a lake or walking through a puddle in your sandals. The bacteria doesn't have teeth. How does it get in?
It doesn't need teeth. It has those corkscrew flagella we talked about.
The drill bit.
Right. It enters the human body through cuts, even microscopic abrasions on your foot that you wouldn't even notice.
Oh, wow.
And furthermore, it can penetrate waterlogged skin.
Like when your fingers get pruned?
Exactly. Think about how your skin gets soft and wrinkled after being in the bath for a long time. That softened tissue is literally an open door for a microscopic drill bit.
That is horrifying.
And it gets worse.
Right.
It can penetrate entirely intact mucous membranes.
So no cut required?
No cut required.
Yeah.
If contaminated water splashes into your eyes, up your nose, or if you accidentally swallow a mouthful while you're swimming,
Game over.
the bacteria can bore directly through the mucosal lining right into your bloodstream.
Okay, so historically, stepping on that biological landmine was mostly an occupational hazard.
Right, like we talked about with Adolf Whale's coal miners.
Yeah, and today it's still a huge risk for, farmers working in flooded rice paddies or sanitation workers in city sewer systems. But the sources we reviewed point to a massive demographic shift happening right now.
Yes.
The landmine is increasingly being stepped on by people just seeking recreation.
The rise of adventure sports and ecotourism has completely changed the epidemiology of this disease, especially in the developed world.
Let's look at that Springfield Triathlon case study from 1998 in the United States, because it perfectly illustrates this shift.
It really is a textbook example.
So you have hundreds of elite athletes gathered in Illinois, and shortly before the race, the area experiences extremely heavy rains.
Typical summer storm.
Right. The athletes do the swimming portion of the race in Lake Springfield, and in the weeks that followed, the local hospitals just started seeing this influx of very, very sick people.
And the epidemiological detective work on that outbreak was intense.
I bet.
The CDC eventually got involved and confirmed that 98 of those athletes had fallen ill with leptospirosis.
98 athletes from one swim.
Yeah. The heavy rains had basically washed animal waste from all the surrounding agricultural lands and wildlife habitats directly into the lake,
Spiking the bacterial load?
massively spiking it in the water right before the athletes jumped in.
And it wasn't an isolated incident either. Look at the Eco Challenge Adventure Race in Borneo in 2000.
Oh, that one is famous in the infectious disease world.
These are some of the toughest endurance athletes on the planet. They were swimming in the Sagama River as part of the course, and the post-race data showed that a staggering 42% of the athletes who were contacted met the clinical case definition for leptospirosis.
Nearly half the field was taken down
Half? Why was it so high?
because they were essentially swimming in the absolute perfect environment for transmission.
Right.
They were in warm, tropical, fresh water. They were totally exhausting themselves. They were sustaining minor cuts and scrapes on rocks in the river, and they were constantly submerging their heads.
So their physical defenses were compromised, and the environmental bacterial load was incredibly high.
A perfect storm for infection.
But, you don't actually have to fly to Borneo to be at risk anymore. And this is where the sources raise a massive red flag about our future.
Yes?
Extreme weather and climate change are fundamentally rewriting the map for leptospirosis.
Climate change is really the engine accelerating this disease globally right now.
How so?
Well, we are seeing heavier, more concentrated rainfall and far more frequent severe flooding events.
Right.
So think about an urban environment like a major city. You have large populations of rats living in the sewers and alleyways, constantly shedding bacteria.
And normally that bacteria might just stay contained down in the sewer.
Exactly. But when a massive flood hits a city, It washes all of that concentrated, highly contaminated urine up out of the sewers and spreads it across human living spaces.
The floodwaters literally weaponize the local environment.
Precisely. We're seeing the geographic footprint of leptospirosis expand significantly.
It's moving.
It is no longer a disease you only get if you travel to a tropical rainforest. Following major hurricane and storm events, we are seeing significant outbreaks in temperate zones now, like Europe and the continental United States.
The boundaries between animal habitats and human spaces are just being washed away.
Literally washed away.
Okay, let's follow the timeline of an actual infection, because this is where the central mystery, how this curable bacteria actually kills, starts to get solved.
Let's trace it.
So you have an athlete swimming in a flooded lake, or a resident walking through a flooded street in sandals. The corkscrew bacteria finds a tiny cut on their waterlogged foot or splashes into their eye.
Okay.
It burrows through the tissue and drops right into the human bloodstream. What happens the exact moment it gets inside?
Well, this brings us to the pathogenesis.
The actual biological warfare happening inside the host.
Exactly. And this is where the biology of Leptospira becomes truly unique and, frankly, incredibly deceptive.
We need to talk about the human immune system's frontline security here, because they fail spectacularly, don't they?
They fail completely. In the human innate immune system, we have these specialized protein receptors on our cells called toll-like receptors, or TLRs.
TLR.
Right. They are essentially biological security guards.
Okay.
Their entire job is to patrol the body, looking for specific molecular patterns that belong to known pathogens. And one of the most important guards is called TLR4.
TLR4. Okay, so when I was reading this, it helped to picture the immune system like a high-security office building.
Like that.
And TLR4 is the guard sitting right at the front desk in the lobby.
Perfect.
This guard has a massive binder full of photos of known criminals. And in the biological world, TLR4's specific job is to look for the lipopolysaccharides, the LPS, of gram-negative bacteria.
Exactly. He's looking for those specific uniforms.
Right. So when a standard gram-negative bacteria, like, say, E. coli or Salmonella, walks into the lobby,
The guard checks the binder, sees the match, hits the massive red alarm button, and the immune system just swarms the lobby and crushes the invasion instantly.
that is exactly how it is supposed to work. But remember when we talked about how Leptospira has a totally unique uniform?
Yes.
Its LPS is structurally distinct from almost every other gram-negative bacteria on Earth.
Right. It doesn't match any of the photos in the human TLR4's binder.
None of them.
Human TLR4 very poorly recognizes the LPS of Leptospira. It is a profound and often fatal biological mismatch.
This just blew my mind when I read it, because earlier you mentioned that mice and rats are basically immune to the severe disease.
They are,
Why?
because mouse TLR4 is structurally different from human TLR4.
So the mouse security guard has the updated photo.
Exactly. When the bacteria enters a mouse, the guard hits the alarm immediately, and the mouse's immune system keeps the infection localized to the kidneys.
But in humans...
In humans, the security guard looks right at the Leptospira bacteria, checks the binder, and basically says, "You don't look like a threat. Go right on through to the elevators."
That is terrifying. It literally just walks right past our primary defense mechanism.
Right past it.
And because there is no alarm, the bacteria has free reign. It begins massive, unchecked replication in the bloodstream.
The medical term for this is bacteremia, or leptospiremia.
And while it is multiplying by the millions, it isn't just floating there passively, is it?
No, it is actively invading.
Yeah.
The bacteria uses specific surface lipoproteins on its outer membrane to grab hold of human tissue.
Okay.
One of the most heavily studied ones is called LipL32.
How does LipL32 actually work?
Think of it like a grappling hook.
A grappling hook.
Yeah, the bacteria uses LipL32 to bind directly to the extracellular matrix of our tissues. That's the structural scaffolding of our organs, things like fibronectin and collagen.
Okay, so it hooks on.
Once it grapples on, it uses that corkscrew motor we talked about to bore deep into the organ tissue itself.
It's drilling right in.
It invades the liver, the kidneys, the lungs, the heart.
Yeah.
And it is even capable of corkscrewing its way right through the blood-brain barrier into the central nervous system.
And it does all of this while the security guard at the front desk is still, like, sipping coffee.
Completely oblivious.
But the human body isn't completely defenseless forever. Eventually, the immune system has to realize there is a massive biological army tearing apart the organs, right?
It does, but by the time it wakes up, it's often way too late for a measured, controlled response.
What happens?
Eventually, A different security guard, another receptor called TLR2, detects the overwhelming massive amounts of leptospiral lipoproteins circulating everywhere in the body.
Okay, so the alarm is finally pulled.
The alarm is pulled, but
Gotcha.
because the bacterial load is already so impossibly high, the immune response completely panics.
It freaks out.
It loses all regulation. It initiates what we call a cytokine storm.
A cytokine storm. We've heard that term a lot in recent years. What is it exactly?
Cytokines are chemical messengers. Under normal circumstances, they tell the immune system where to go and how hard to fight.
This direct traffic.
Right. But in a cytokine storm, the body just dumps massive, dysregulated amounts of pro-inflammatory cytokines into the blood all at once. Specifically, chemicals like IL-6 and TNF-alpha.
What do IL-6 and TNF-alpha actually do to the body in those massive quantities?
They're essentially chemical alarm bells, screaming at all the blood vessels to open up and let white blood cells through to fight the infection.
Okay, that sounds helpful.
It is, normally.
Yeah.
But when there is way too much TNF-alpha, it causes severe capillary vasculitis.
Vasculitis, meaning inflammation of the vessels.
Exactly. The cells lining your blood vessels, the endothelial cells, become massively swollen and damaged. They actually undergo necrosis, which is premature cell death.
Oh, wow.
The junctions between the cells break apart, and the blood vessels literally start to leak massive amounts of fluid and eventually blood directly into the surrounding tissues.
So your plumbing is just falling apart.
Yes. And to make matters worse, the sources note that the body paradoxically releases high levels of an immunosuppressive cytokine called IL-10 at the exact same time.
Wait, immunosuppressive? Why would it do that?
It's total chaos. The body is hitting the gas pedal and the brake pedal simultaneously.
That makes no sense.
It's releasing chemicals to cause massive inflammation, while also releasing IL-10 to suppress the immune system, resulting in total physiological breakdown.
This right here is the most crucial takeaway of the pathogenesis, I think. The most severe, life-threatening damage in leptospirosis isn't actually caused by the corkscrew bacteria eating your tissue.
No.
It is caused by your own immune system tearing up your body's vascular plumbing in a desperate attempt to kill an invader that it allowed to multiply out of control in the first place.
Which perfectly explains the classic biphasic or two-phase nature of the disease.
Let's talk about those two phases.
In the first phase, the septicemic phase, the bacteria is quietly multiplying in the blood, slipping past the immune system.
That's week one.
Right. Then in the second phase, the immune phase, your own antibodies, and the resulting cytokine storm cause all the catastrophic organ damage.
And that internal vascular destruction, all those leaking pipes all over the body, translates directly to the chaotic clinical battlefield that doctors see in the emergency room.
It does. It explains the completely bewildering multi-organ symptoms that make leptospirosis such a master of disguise.
Because it mimics almost every other endemic or tropical fever out there, doesn't it?
Almost all of them. Dengue, malaria, typhoid.
So after a person is exposed, the incubation period is usually, what, 5 to 14 days?
Usually 5 to 14 days, though it can be up to a month.
So you are literally walking around feeling perfectly fine while the bacteria multiplies into the millions.
Yes. And then the disease kicks off abruptly.
It hits you like a truck.
It doesn't build slowly. Patients present with a sudden, very high fever, severe chills, and an intense frontal headache.
But there's an early clinical clue that doctors are trained to look for, right? The pain.
Yes, the pain is a major indicator. Patients develop highly characteristic agonizing myalgia muscle pain, specifically in the calves and the lower back.
Why the calves? What is the bacteria actually doing to the leg muscles?
The bacteria directly invade the muscle fibers themselves, and the resulting inflammation causes what we call non-purulent myositis. The tissue becomes incredibly swollen and tender. In severe cases, it progresses to rhabdomyolysis.
Rhabdo, I've heard of that with extreme athletes.
Right, it's a condition where the skeletal muscle tissue literally breaks down and dies rapidly.
Just melting away.
And it leaks muscle proteins into the blood, which can cause intense pain and further damage the kidneys.
The sources highlight another major pathognomonic clue, too. A symptom specifically characteristic of this disease that helps differentiate it from, say, just a bad flu.
Ah, conjunctival suffusion.
Yes, conjunctival suffusion.
It is a critical diagnostic marker. It presents as intensely bright red eyes.
Like pink eye.
No,
Yeah.
and that's the key distinction. Unlike typical pink eye or bacterial conjunctivitis, there is no pus.
No pus.
No crusting, no inflammatory exudate at all. The eyes are just violently, clearly red.
Why are they so red if there's no infection on the surface of the eye itself?
It's a direct visual manifestation of the capillary vasculitis we just discussed.
The leaking pipes again?
Exactly. The tiny blood vessels in the whites of the eyes, the conjunctiva, are dilated, inflamed, and leaking due to the cytokine storm. You are quite literally seeing the internal vascular destruction happening in real time.
That is so eerie. So what we just described, the fever, the severe calf pain, the red eyes, that represents the mild anecteric form of the disease, right?
Correct. It accounts for about 90% of symptomatic cases. It's miserable, but it's usually self-limiting.
It's that remaining 10% where the disease really earns its terrifying reputation.
Yes.
This is where we return to 1886. We return to Weil's disease, the severe icteric form.
Weil's disease is the classic severe presentation, and it is characterized by a very specific triad of organ failure.
What's the triad?
Severe jaundice, acute kidney injury, and hemorrhage.
Let's break down the jaundice first, because the mechanism here is fascinating, and it's very different from what people might assume.
It is.
Because when people hear jaundice, they immediately think of liver failure, like viral hepatitis, where the liver cells are actively dying.
Right. In viral hepatitis, you have massive hepatocellular necrosis.
The cells are dying.
The liver cells themselves are being destroyed en masse, which causes bilirubin, the yellow pigment, to build up in the blood.
But Leptospira operates differently.
Very differently. The bacteria don't necessarily kill the liver cells in massive numbers. Instead, they use that corkscrew motion to invade the microscopic spaces between the liver cells, the hepatocytes.
They attack the plumbing again.
Exactly. They attack and disrupt the tight intercellular junctions.
Okay, so normally liver cells process bilirubin into bile and secrete it into these tiny ducts to be carried away to the gallbladder, right?
Right. But because the bacteria have torn apart the junctions between the cells, the bile simply leaks out of the ducts and spills directly back into the bloodstream.
Which gives the patient a profound, deep, bronze-hued jaundice.
Yes.
And while the liver plumbing is actively leaking, the kidneys are taking a massive beating at the same time.
The kidney damage is profound. The bacteria directly target and colonize the proximal tubules of the kidney.
What do those do?
That's the area responsible for reabsorbing essential nutrients and electrolytes back into the blood. This causes a very specific and highly dangerous type of kidney failure: non-oliguric acute kidney injury with hypokalemia.
Okay, let's translate that for the listener.
Sure.
Non-oliguric basically means the patient might still be producing plenty of urine, right?
Right.
Which can completely trick a doctor into thinking the kidneys are fine.
It's a huge trap.
But hypokalemia means the urine they're producing is doing something really dangerous.
Hypokalemia means dangerously low potassium levels in the blood.
Okay.
Because the proximal tubules are damaged, the kidneys lose the ability to hold on to potassium. They essentially waste it, dumping massive amounts of potassium out of the body through the urine.
Why is that a distinguishing factor?
Because in many other forms of acute kidney injury, the kidneys stop working entirely and potassium levels spike dangerously high.
Oh, I see.
But in leptospirosis, they plummet early in the disease, which can cause fatal cardiac arrhythmias.
Wow. But even Weil's disease isn't the most lethal manifestation, is it?
No, it's not.
If we are looking for the absolute worst case scenario, we have to talk about leptospirosis-associated pulmonary hemorrhagic syndrome, or LPHS.
Yeah. When clinicians discuss LPHS, they do so with immense respect and fear.
It's that bad.
We are talking about a clinical presentation with a fatality rate exceeding 50%, even in modern, well-equipped intensive care units.
Remember the triathlete from the very beginning of our deep dive whose lungs filled with blood?
This is LPHS.
What is mechanically happening in the lungs here?
Is the absolute extreme end of that cytokine-driven vasculitis.
The inflammation.
Right. The capillaries wrapping around the alveoli in the lungs, the microscopic air sacs where oxygen is transferred into the blood, are damaged so severely that they simply rupture.
Oh my God.
The alveoli literally fill with blood. The patient loses the ability to exchange oxygen. They're drowning from the inside out.
And the most terrifying aspect of LPHS is that it doesn't even play by the rules.
It really doesn't.
It often happens rapidly between days 4 and 6 of the illness. And crucially, it can occur without the patient ever developing the classic jaundice of Weil's disease.
Which makes it an absolute nightmare to diagnose. A patient might come into the emergency room presenting with what looks like severe pneumonia or acute respiratory distress.
And a doctor isn't necessarily thinking of a waterborne spirochete.
Not at all. And then suddenly the patient is suffering massive hemoptysis,
Coughing up blood.
copious amounts of blood.
Though the sources warn that in some insidious cases, the hemorrhage is entirely alveolar, right?
Yes.
Meaning the bleeding stays deep in the tiny air sacs, the lungs fill up like a sponge, and the patient might never cough up a single drop of blood until their oxygen levels just plummet to fatal levels and they code.
That deceptive presentation is exactly why the 1995 outbreak in Nicaragua was so baffling to global health experts.
What happened there?
Well, following heavy rains and flooding, rural populations were coming into clinics suffering from rapid, fatal respiratory failure.
And what did the experts think it was?
The CDC and the WHO initially thought it was an outbreak of hantavirus, or perhaps a highly virulent strain of dengue fever, because the patients lacked the classic Weil's disease jaundice.
It was a total mystery.
It took weeks of epidemiological investigation to finally realize they were dealing with pulmonary leptospirosis.
So the presentation is a total chameleon. It can look like dengue, malaria, typhoid, hantavirus, even a bad flu.
Exactly.
So how does a doctor in a busy emergency room actually figure it out before it's too late?
If the clinical picture is blurry, doctors have to look for the diagnostic triad in the standard blood work.
Okay, what is that?
Well, if you connect the dots of the pathophysiology we just covered, the routine lab results actually scream leptospirosis.
What are the 3 points of the triad?
First, you look at the liver panel. You will see a massively high bilirubin level because the bile is leaking into the blood.
Right, the broken plumbing.
Exactly. But you'll only see mildly elevated liver transaminases, like AST and ALT.
Why is that important?
Because if it were viral hepatitis, the AST and ALT would be sky high, because the liver cells are dying en masse.
Okay, that's point 1. High bilirubin, low astalt. What's point 2?
Second, you look for highly elevated muscle enzymes, specifically CPK. That enzyme leaks into the blood from the severe muscle damage happening in the calves.
Which is calf damage you don't see in dengue or hepatitis.
Exactly. And third, you look at the kidney panel for that hypokalemia we discussed.
The low potassium.
Right, the low potassium combined with acute kidney injury. If you have a patient with fever and red eyes, and you see high bilirubin with low astalt, high muscle enzymes, and low potassium,
You must immediately suspect leptospirosis.
you have to.
But suspecting it and definitively proving it are 2 very different battles.
Very different.
Because the definitive diagnostic tests for this disease are notoriously difficult, creating this incredible high-stakes race against time. Which brings us to the diagnostic ticking clock.
Diagnosing leptospirosis requires the physician to know exactly where the bacteria is hiding in the body on any given day. You cannot use the same test on day 3 that you would use on day 12.
We have to go back to the biphasic nature of the disease then, week 1 versus week 2.
Exactly. In the first 5 to 7 days, the leptospiremic phase, the bacteria are actively multiplying in the bloodstream, trying to find tissues to invade.
Okay.
So during this brief early window, a PCR test on the patient's blood is highly effective. PCR amplifies and detects the actual DNA of the bacteria.
But the clock is ticking loudly. Because right around day 7, the immune phase begins.
Right.
The body starts churning out massive amounts of IgM antibodies. And these antibodies sweep through the blood and clear the bacteria out of the circulation.
The bacteria flee the bloodstream and hide deep in the organ tissues in the urine.
So if a patient comes in on day 10, severely ill with Weil's disease, and the doctor orders a PCR test on their blood, what happens?
It will almost certainly come back completely negative.
Because the bacteria are gone from the blood.
Exactly. And the doctor might see a negative test and incorrectly rule out leptospirosis while the patient continues to deteriorate from the immune-mediated organ damage.
It's basically like investigating a crime scene.
How do you mean?
Well, in week 1, the burglar is actively running around inside the house. So you use a PCR test to catch the burglar in the act in the living room, which is the blood.
Okay, I see where you're going.
But by week 2, the burglar has fled the living room and is hiding down in the sewer, which is the urine. You can't find him in the house anymore.
Right.
So instead of looking for the burglar himself in the blood, you have to look for the footprints, the broken windows, and the tripped alarms he left behind.
That is an excellent analogy. In week 2, you stop looking for the bacteria's DNA in the blood, and you start looking for the evidence that the immune system is actively fighting.
The broken windows.
Right. You use an IgM ELISA test, which detects the early antibodies the body produces. And you can also run a PCR on the urine, hoping to catch the genetic material of the bacteria as it sheds from the kidneys.
Now, in reading the sources, I came across the historical gold standard for diagnosing leptospirosis. It's called the microscopic agglutination test, or MAT.
Ah, the MAT.
But honestly, reading how this test is actually performed sounded like an absolute logistical nightmare.
It is incredibly complex, which is exactly why it is almost exclusively performed in highly specialized national reference laboratories, like the CDC.
It is not something your local hospital can just run.
Definitely not.
What does the test actually require them to do?
It requires a laboratory to maintain massive panels of live, highly motile, dangerous Leptospira bacteria.
Live bacteria.
Yes, we are talking about keeping dozens of different living ceravars, all those different uniforms we talked about, alive and cultured at all times.
So they just have racks of live corkscrewing bacteria waiting in tubes in a lab.
Yes, they do. And to run the test, the scientist mixes the sick patient's blood serum with these various live bacterial cultures.
Okay, and then what?
Then they look at the mixture under a specialized dark field microscope. If the patient has leptospirosis, their serum will contain antibodies.
Right.
When those antibodies hit the live bacteria in the tube, they latch onto them and cause the live bacteria to clump together, or agglutinate.
So the scientists literally watch the bacteria clump up under the microscope to prove the patient has the disease.
Exactly.
But that takes a massive amount of time, doesn't it?
It does. It is not a rapid point-of-care test by any means. To be definitively diagnostic, MNP usually requires taking an acute blood sample early in the disease and then a convalescent sample a few weeks later, looking for a fourfold rise in the antibody levels.
So it's brilliant science, and it confirms the diagnosis and the specific cerevar after the fact.
Yes.
But it is entirely too slow to guide the critical initial treatment decisions an emergency room doctor has to make on day one.
It is entirely retrospective in most critical cases.
And what happens when all of these tests, PCR, ELISA, MAT, what happens when they all fail but the patient is actively dying?
Yeah.
The sources highlight a truly incredible landmark case study from the New England Journal of Medicine in 2014 that answers this.
Oh, that is a phenomenal case.
Yeah.
It really pushed the boundaries of modern diagnostics.
It involved a 14-year-old boy who presented to a hospital with just a devastating clinical picture.
Right.
He had massive brain swelling. He was locked in status epilepticus, which is continuous severe seizures, and he was placed in a medically induced coma just to save his brain.
And for 4 months, a team of top-tier specialists ran exhaustive diagnostic workups.
4 months.
They ran every infectious disease panel imaginable. They even performed invasive brain biopsies.
But every single test, including blood PCR, and serology for leptospirosis, came back completely negative.
They were watching him slowly die and had zero idea why.
The compounding tragedy for this young man was that he suffered from severe combined immunodeficiency, or SCID.
Right, his immune system was fundamentally broken.
Because he couldn't mount an immune response, he wasn't producing the IgM antibodies that standard tests like ELISA and MAT rely on to detect the disease.
His body wasn't leaving any footprints for the doctors to find.
So how did they finally find the microscopic drill bit?
The breakthrough only came when the clinical team decided to utilize unbiased next-generation sequencing, or NGS, on a sample of his spinal fluid.
Okay, what does that actually mean?
Instead of testing for specific diseases one by one, NGS takes a sample and sequences every single piece of DNA inside it. Human, viral, bacterial, fungal, everything.
They just dumped the entire genetic soup of his spinal fluid into a supercomputer.
Exactly. And they generated over 3 million genetic reads from that tiny sample.
3 million.
And out of those 3 million reads, the computer flagged just 475 reads that did not belong to a human.
Oh, wow.
Those 475 tiny fragments mapped perfectly to the genomic blueprint of Leptospirosis enterosi.
They found a microscopic genetic needle in a 3 million piece haystack.
They did.
And the moment they found it, they knew exactly what to do. They started targeted IV antibiotics, and that 14-year-old boy, who had been in a coma for months, woke up and walked out of the hospital 32 days later.
It's a miraculous story of medical persistence and the power of genomic sequencing.
It really is.
But from a global public health perspective, it highlights a massive, glaring limitation.
Right, because next-generation sequencing costs thousands of dollars, requires supercomputers, and takes days to run.
It is a cutting-edge research tool.
It is absolutely not available in a rural clinic treating a farmer in Southeast Asia, or even in most standard urban emergency rooms in the U.S.
Not at all. Which leads us directly to the most crucial, universally agreed upon clinical maxim across every single guideline and source we reviewed for this deep dive.
What's the rule?
Never, ever wait for laboratory confirmation to start treatment.
The golden rule. If it looks like a duck and quacks like a duck, you treat it for leptospirosis immediately.
I cannot stress that enough. If the clinical suspicion is there, if you have a patient presenting with high fever, severe calf pain, conjunctival suffusion without pus, and they have a history of freshwater exposure, flooding, or rodent contact,
You pull the trigger.
you do. Delaying antibiotics for even 24 hours while waiting for a PCR or an ELISA result to come back from the lab can be the difference between a full recovery and fatal pulmonary hemorrhage. You must initiate treatment presumptively.
So once that clinical suspicion triggers action, how do we actually fight back against this corkscrew invader?
With antibiotics primarily.
We know it's a gram-negative bacteria with heavy armor. What is in the medical arsenal?
The specific choice depends entirely on the severity of the clinical presentation. For mild anapteric disease, the patients who are miserable but stable enough to be managed in an outpatient setting, oral doxycycline is the undisputed drug of choice.
Doxycycline is a classic, really versatile antibiotic.
It is highly effective against Leptospira, and it offers a massive strategic advantage in tropical or rural settings. It also effectively treats competing diagnoses.
Oh, that's clever.
Right. If the doctor isn't perfectly sure if the patient has leptospirosis or scrub typhus or murine typhus, all of which can present with similar fevers, and myalgia doxycycline covers all of them.
That is a huge advantage. Are there alternatives if they can't take doxy?
Yes. If doxycycline is contraindicated, for instance, in young children or pregnant women where it can affect bone and tooth development, alternatives like amoxicillin or azithromycin are utilized.
But for severe disease, say, a patient hospitalized with Weil's disease, failing kidneys, jaundice, or the beginnings of LPHS oral pills aren't going to cut it, you have to bring out the heavy artillery.
You do. In those severe hospitalized cases, the guidelines recommend immediate administration of intravenous antibiotics.
Like what?
The gold standard has long been IV benzyl penicillin, also known as penicillin G, given in massive doses every 6 hours.
Okay.
Or ID ceftriaxone, a third-generation cephalosporin, is equally effective and only needs to be dosed once a day, which is often much easier for the nursing staff.
These 5E antibiotics aggressively attack the bacteria, ripping apart their cell walls.
Yes.
But the sources detail a severe, paradoxical, and honestly terrifying risk to administering these life-saving drugs. It is called the Jarisch-Herxheimer reaction, or JHR.
The Jarisch-Herxheimer reaction is a very well-documented, volatile physiological phenomenon.
When does it happen?
It occurs frequently within the first 24 hours of starting antibiotic therapy for any spirochetal infection, including leptospirosis, syphilis, and Lyme disease.
Okay, I read the description of this, and it sounds absolutely counterintuitive.
It does seem that way.
You are telling me that a doctor gives a patient the exact medicine required to save their life, and the immediate result is a massive biological explosion that makes the patient look and feel substantially worse, often pushing them right to the brink of death.
Yes, and understanding why it happens is critical for anyone managing these patients.
Why does it happen?
When those massive doses of IV penicillin hit the bloodstream, they don't just kill a few bacteria slowly. They cause massive, rapid, systemic bacterial cell death.
Millions of microscopic drill bits exploding all at once.
Precisely. As millions of Leptospira cells rupture simultaneously, they suddenly dump all of their internal toxins and surface lipoproteins, like that LipL32 grappling hook we discussed earlier, directly into the patient's bloodstream in one massive wave.
And the immune system security guards, those TLR receptors, see this unprecedented flood of bacterial debris and absolutely panic.
The immune system reacts as if the infection just multiplied 1,000-fold in a single instant. It triggers a massive acute inflammatory spike.
What does that look like in the hospital bed?
Clinically, the patient will experience a sudden, extreme spike in fever, violent rigors, and uncontrollable shaking chills. They will develop tachycardia, a dangerously fast heart rate, and most critically, a profound drop in blood pressure.
Why does the blood pressure drop so dangerously?
Because that massive sudden release of cytokines forces all the blood vessels in the body to dilate, to widen all at once. When all the pipes in the system suddenly get wider, the pressure drops instantly. There isn't enough pressure to pump blood to the brain and vital organs. It clinically mimics severe septic shock or a rapid, fatal deterioration of the disease itself.
I can completely imagine a nurse or a junior doctor seeing this happen an hour after hanging an IV bag of penicillin and immediately thinking, oh no, they're allergic to the drug. I need to stop the infusion right now.
That is exactly the greatest danger of JHR misinterpreting it.
Right.
Stopping the antibiotics would be a catastrophic mistake because it would allow the remaining bacteria to rebound and continue destroying the organs.
Wow.
JHR is not a drug allergy like anaphylaxis. It is violent, chaotic proof that the drug is working exactly as intended.
So how do they get the patient through it?
The management requires aggressive, proactive, supportive care, almost exclusively in an intensive care unit setting.
They just have to weather the storm.
The clinical team has to push through the storm. They use intravenous fluids and vasopressor medications to force the blood vessels to constrict, mechanically supporting the blood pressure until the inflammatory wave passes. They aggressively manage the fever.
Which proves a major point raised in all the clinical guidelines. The critical care support is just as important, if not more important, than the antibiotics themselves.
Absolutely.
A patient with severe Weil's disease or LPHS doesn't just need a pill. They need a machine to keep them alive while the drugs work.
The ICU interventions required for severe leptospirosis are immense.
What else do they need?
Well, if the bacteria and the cytokine storm have destroyed the proximal tubules of the kidneys, the patient will develop profound acute kidney injury. They require early and aggressive hemodialysis.
So the machine does the work of the kidneys.
The dialysis machine has to take over, clearing the deadly buildup of toxins and strictly managing those fluctuating potassium levels to prevent cardiac arrest.
And what if the patient develops LPHS? What if their lungs are actively filling with blood?
Then they require immediate lung protective mechanical ventilation. A tube is placed in their airway, and a machine forces oxygen into the lungs using high pressure to keep those bleeding alveoli open and functioning.
But the case studies mention scenarios where even a ventilator isn't enough.
Yes.
If the alveolar hemorrhage is so massive that the lungs are completely flooded, the ventilator is just pushing air into a pool of blood. No oxygen is making it into the bloodstream at all.
In those absolute most desperate, dire cases of pulmonary hemorrhage where the lungs have completely failed, advanced hospitals have successfully utilized the technology called VV-ECMO.
Extracorporeal membrane oxygenation.
Exactly. It is the highest level of life support available. Large cannulas are inserted into the patient's major veins.
And it does what?
The ECMO machine physically pumps the deoxygenated blood out of the patient's body, runs it through an artificial membrane that removes the carbon dioxide and infuses it with oxygen, and then pumps the vitalized blood back into the body.
It literally bypasses the lungs entirely.
It does, giving the alveolar tissue time to stop bleeding and heal while keeping the brain and organs alive.
It is truly incredible what modern medicine can do to pull someone back from the brink of this disease.
It is.
But the sources also emphasize that certain populations are at a significantly higher risk of complications, and we need to highlight the danger to pregnant women.
Leptospirosis during pregnancy is a terrifying clinical scenario for two main reasons.
Okay, what's the first?
First, the symptoms, the fever, the liver enzyme abnormalities, the dropping platelet counts, they clinically mimic severe, life-threatening obstetric complications, specifically HELLP syndrome.
HELLP syndrome.
Yes. HELLP stands for hemolysis, elevated liver enzymes, and low platelets.
So an OBGYN might look at the blood work and think the pregnancy itself is causing the organ failure, completely missing the fact that it's an infectious disease requiring antibiotics.
Exactly, leading to fatal delays in treatment.
And the second reason?
Second, the Leptospira bacteria is incredibly invasive. We know this.
Right, the corkscrew.
It easily crosses the placental barrier. The infection carries a tragically high risk of intrauterine infection, leading to fetal demise, spontaneous miscarriage, or stillbirth.
That's devastating.
It requires rapid, multidisciplinary intervention between infectious disease specialists and maternal-fetal medicine teams.
Given how devastating and complex this pathogen is once it gets inside, naturally, the ultimate goal is to never let it breach the skin in the first place.
Prevention is key.
How do we prevent leptospirosis? The sources mention a strategy called chemoprophylaxis.
Chemoprophylaxis involves taking a preventative dose of antibiotics, usually a weekly dose of doxycycline, before and during an exposure to a high-risk environment.
To kill the bacteria before it can establish an infection.
Exactly.
Like taking anti-malarial pills before going to a jumble. I would assume every triathlete or ecotourist should just pop a doxycycline a week before a race, right?
You'd think so.
But the evidence on its effectiveness seems surprisingly mixed for the general public.
The evidence is highly nuanced. Chemoprophylaxis gained massive popularity after a landmark 1984 clinical trial involving U.S. soldiers deployed for jungle training in Panama.
Did it work for them?
It did. The soldiers taking weekly doxycycline had a vastly lower infection rate than the placebo group. It proved highly effective for healthy adults in short-term, predictable, high-risk exposures.
But when they tried to roll that strategy out to civilian populations, it didn't work as well.
Correct. Subsequent studies, particularly in post-flood community settings in endemic regions like the Andaman Islands, showed that mass distribution of doxycycline didn't reliably prevent all infections or halt outbreaks in the general population.
Why not?
Compliance is a major issue, people forget to take it, and the drug just isn't a magical shield. Today, chemoprophylaxis is generally reserved for specific groups.
Like military personnel on deployment.
Right, or outbreak response workers, or athletes in adventure races.
It is not a substitute for standard physical precautions.
Absolutely not.
The public health manuals emphasize basic barriers. Covering open cuts with waterproof medical dressings before swimming.
Wearing protective rubber boots if you have to wade through floodwaters or work in agriculture.
And aggressively controlling the rat population in urban centers to reduce the environmental contamination at the source.
Environmental modification and physical barriers are far, far more effective on a population level than just handing out preventative antibiotics.
But what about a vaccine? I kept waiting to read about a vaccine. We have flu shots. We have tetanus shots. We have vaccines for countless pathogens. Surely, for a disease that infects 1 million people a year and kills 60,000, we have a vaccine.
This is perhaps one of the greatest ongoing frustrations in the entire field of tropical medicine.
There isn't one.
There is no widely available, FDA-approved human vaccine for leptospirosis in the United States or the United Kingdom.
Why not? We know exactly what the bacteria looks like.
It goes right back to the taxonomy discussion we had earlier. Remember the 300 different serovars?
The 300 different uniforms.
Exactly. When you create a vaccine using an inactivated Leptospira bacteria, it only provides immunity against that specific uniform.
Oh, I see the problem.
If a pharmaceutical company creates a vaccine using Cerovar canicola, it provides zero protection if you step in a puddle contaminated with Cerovar pneumoniae.
Because the immune system is looking for the wrong outfit again.
The genetic diversity of the surface antigens makes creating a universal human vaccine incredibly difficult.
So we just don't have one at all.
A few countries, like Cuba and France, do utilize whole cell inactivated vaccines for certain high-risk workers. But they require multiple doses, they provide only short-term immunity, and crucially, they are formulated only to protect against the specific serovars historically prevalent in those localized regions.
So they aren't useful globally.
Not at all.
But interestingly, the sources note that veterinary medicine is light years ahead of human medicine here.
They really are. The leptospirosis vaccine is considered a core routine requirement for domestic dogs in many parts of the world.
My dog gets one every year.
Right, and the veterinary vaccines are multivalent. They actually combine the 4 most common serovars that infect dogs into one shot.
So we can protect our golden retrievers from the puddles in the park, but we can't easily protect the humans walking them.
Exactly. Vaccinating dogs is crucial for their health, but it doesn't solve the human problem because dogs aren't the primary environmental reservoir driving human outbreaks.
The rats and livestock are.
And we cannot practically vaccinate millions of wild rats in the sewers.
Which perfectly frames the massive scale and the fundamental ecological complexity of this whole issue. Let's take a moment to really summarize the incredible journey we've taken today, because tracing the path of this pathogen reveals exactly why it is so uniquely dangerous.
It is a staggering biological cascade from start to finish.
We started with a microscopic, tightly coiled bacterium quietly hiding out in a state of evolutionary truce inside the kidney of an asymptomatic city rat. That rat sheds the bacteria into a puddle in an alleyway. Heavy, climate-driven rains, which are, becoming more frequent every single year, wash that puddle into a local recreational lake or flood the city streets.
And then you, perhaps an outdoor enthusiast, a triathlete, or just a resident walking home in the rain, come into contact with that water.
The bacteria uses its unique corkscrew motility to breach your waterlogged skin or your mucous membranes. It slips right past your innate immune system because your TLR4 security guards just don't recognize its unique outer uniform.
It multiplies rapidly in your blood, grappling onto your tissues with surface proteins, boring into your liver, your kidneys, and your lungs.
And when your immune system finally wakes up to the massive invasion, it completely overreacts. It launches a dysregulated cytokine storm that ravages your own vascular system.
This leads to the hallmark capillary vasculitis. Your blood vessels leak, leading to the bronze jaundice of Weil's disease, the dangerous potassium-wasting failure of your kidneys, or, in the most terrifying cases, LPHS, where your lungs just fill with blood.
Meanwhile, the doctors are forced into a high-stakes race against a shifting diagnostic clock, trying to decipher a chameleon-like clinical presentation that mimics a dozen other tropical diseases.
Because by week 2, the bacteria has fled the blood, rendering standard PCR tests entirely useless, and forcing labs to hunt for antibodies or rely on massive supercomputers to sequence spinal fluid.
And ultimately, the clinical team has to fight back with heavy intravenous antibiotics, fully knowing that the drugs will likely trigger a violent Jerisch-Herxheimer reaction,
Requiring immense intensive care support dialysis, ventilators, and ECMO just to keep the patient alive while the medicine works.
It is a devastating multi-system biological storm, driven entirely by our interaction with a changing environment. And that is exactly why this matters to you, the listener.
It matters to everyone.
Whether you are an adventure sports enthusiast planning a trip to Borneo, a traveler exploring tropical regions, a farmer working the land, or just someone living in an urban area experiencing increasingly severe weather and flooding, this hidden biological landmine is real.
The threat is no longer confined to distant jungles or 19th century coal mines. It is in our cities, our recreational waters, and literally in our backyards.
Understanding how this pathogen operates, how it hides in asymptomatic animals, how it strikes the human body, and how crucial early recognition is the ultimate shortcut to being well-informed and keeping yourself and your family safe.
If you get sick after a flood or a triathlon, you now know to tell your doctor about the water.
You know not to wait for the lab test.
Exactly. I want to leave you with a final thought today, drawn directly from the One Health approach that was so heavily emphasized in our public health sources.
The One Health approach being the idea that human health, animal health, and environmental health are all intrinsically linked.
Exactly. For centuries, modern medicine has largely operated under the assumption that we can cure human diseases by focusing solely on the human body, creating a better pill, a stronger vaccine, a faster blood test.
Treating the individual.
Right. But leptospirosis proves the limitation of that kind of thinking. The bacteria isn't adapting to hunt us. We are altering the world in ways that force us into its path.
Climate change is forcing the overlap.
If rising global temperatures and increased severe flooding continue to blur the boundaries between human living spaces, animal habitats, and environmental water sources, the global burden of zoonotic diseases like leptospirosis will only continue to grow exponentially.
We can't just ECMO our way out of it.
No, we cannot rely solely on ICU interventions to save us from an increasingly weaponized environment. Perhaps our future medical breakthroughs for diseases like this won't come from a new antibiotic or a more complex diagnostic machine.
Where will they come from?
Perhaps true prevention will require a massive paradigm shift. It will require us to step outside the hospital walls, look at the changing climate, and realize that to truly protect the human host, we're going to have to figure out how to treat the entire ecosystem as the patient.
Treat the ecosystem as the patient. That is a really profound way to look at it. It absolutely changes the way you view the world. It does. It makes you look at that pristine, inviting freshwater lake or that flooded city street just a little bit differently, doesn't it? It might look clear and refreshing on a hot day, but beneath the surface, it is a complex, deeply connected, and sometimes incredibly hazardous biological web. Thank you for joining us on this deep dive into the microscopic world. Stay curious, stay informed, and we will see you next time.